So with this video, we will begin talking about the special senses, and here we go. And this one is going to be about olfaction, the sense of smell, and gustation, the sense of taste. So the special senses, I want to remind you, are all the receptors are all part of specialized sensory organs that are located in the head. And they include the senses of olfaction, gustation, hearing, equilibrium, and vision or sight. And we're going to start by talking about olfaction, the sense of smell. And there are two olfactory organs, and they're located in the epithelial tissues that line the roof of the nasal cavity. So up in the top of the nose, one on the right, one on the left, and that's where we have those olfactory organs. And they're composed of three sets of tissues. There's an olfactory epithelium, and it contains the actual olfactory receptor cells, the supporting cells, the columnar epithelial cells, and these cells all contain the pigment lipofuscin. It's kind of a yellowish brown, and so this epithelial tissue has a yellowish brown color. So we've got the olfactory epithelium, where we actually do the sensing of the odorant molecules. There's another group of cells that are referred to as the lamina propria. This is areolar tissue, and it contains blood vessels, nerve fibers, and the olfactory glands. These olfactory glands are also called Bowman's glands, but again, in the processes of removing, stripping off people's names from structures, they are more properly called now the olfactory glands, and their job is to secrete a really thick yellowish mucus. So lamina propria is just this areolar tissue, provides a pathway for blood and blood vessels and nerve fibers, and also produces a secretion of this yellowish mucus. And then last of all are the olfactory receptor cells. And these are very highly modified bipolar neurons that are embedded there in the columnar epithelium, and they are sensitive to dissolved chemicals, to the odorant molecules. Write that word for you, odorant molecules. Those are the molecules that have the that can be responded to by these olfactory receptor cells. Okay, so three sets of cell types or tissues. There's lamina propria, the connective tissue. There's olfactory epithelium, which are the cells that support the olfactory receptor cells. And then the olfactory receptor cells themselves, which are these very highly modified bipolar neurons. And if you remember bipolar neurons, they have a cell body and then an axon and a dendrite, a single long dendrite and a single long axon. And the only places that we find these bipolar neurons are as sensory cells or sensory receptors. And before we go on too far, these are the only neurons in the body in which the dendrites have direct contact with the external environment. And so the dendrite portion of the olfactory receptor cells they stick out from the epithelium that they're embedded in, and have direct contact with the external environment. And so as air passes across the olfactory membrane, that sensitive region of the nasal passages, then the chemicals that are in the air dissolve in the fluids of the mucus that's secreted by those olfactory glands. There are extensions off of the dendrites. These are referred to as cilia, but if you think back to chapter chapter 3, I guess that was, where we were looking at a cell, we always talked about cilia as being these motile extensions. Well, these are non-motile. They don't move, these cilia. But they extend out into the mucus. And so, the cilia are studded with, the membranes are studded with these receptor proteins that are there to bind the odorant molecules. And we have about 12 million of these receptor cells in between these two olfactory membranes. Okay, so these cilia stick out, and when they receptor proteins bind that odorant molecule, that binding can cause local potential changes in the olfactory receptor cell membrane. And if there's sufficient stimulation, then an action potential will result along the cell's axon, that bipolar neuron axon, and synapse then with interneurons that will carry that message about having encountered this odorant molecule up to the portions of the brain that are busy interpreting that. We'll talk about those in just a minute. So because these olfactory cells are exposed, then they can be easily damaged, and so they have a relatively short lifespan, 30 to 60 days. So 1 to 2 months is about all they can last. They're located pretty far out of the pathway of airflow. And so behaviorally, we've learned to use a sniffing to change the airflow patterns in the nasal passages and help to bring odorant materials in closer contact with that olfactory membrane. And if you get a cold or some other, sick in some way that results in excess mucus production, then those cilia that are sticking out, they can be so embedded in that mucus that's produced that you may have a significant reduction in sensitivity to odors. So it gets harder to smell, and because smell and taste are so closely, they're just very closely tied, we get a lot of information about taste from this olfactory epithelium. When we have a cold, then the tastes of food can be really muted too. Food just doesn't taste as good. Over time, as we age, olfactory cells become less abundant in the olfactory membrane, and the receptors that we do have become much less sensitive than they were when we were young. And so, I'm suggesting that that's why Grandma really sprays that perfume on, is because she's not smelling it the same way that she used to when she was younger. And it takes a little bit more of those odorant molecules in order to really appreciate and get that smell. Okay, so what are the pathways then that the sensory input comes into the brain on? So those olfactory axons, or olfactory receptor cell axons, they pass through very tiny openings in the ethmoid bone that makes up the superior surface of the nasal cavity. And on the far side of that bone, then they join together to form the olfactory nerves. That's cranial nerve number 1, if you remember from old OB. So if you look at figure 12.5 in your book, 12.5 is showing those olfactory receptors cells there with their cilia that are hanging off of them, and you can see that the axon end, they thread together and go up through those very narrow little pores, and then the dendrites, I'm sorry, not the dendrites, but the axon terminals will synapse then with interneurons that are a portion of the olfactory bulb. So all those interneurons that the axons of the olfactory receptor cells, all the interneurons that they synapse with, they make up the substance of the olfactory bulb. And in our sheep brain dissection, you probably had at least a part of, if not a whole bunch of, the olfactory bulb still attached to the brain. So those interneurons, their axons form an olfactory tract that begins there in the olfactory bulb and then leaves the bulb, the olfactory bulb, and terminates in the primary olfactory cortex. So off it goes deeper into the brain to find the cortical region of the brain where where they terminate. Other interneurons also carry information about the smells that we are noticing to the limbic system, to the interneurons that make up the limbic system, that emotional center of the brain. And so there are strong associations with memories and emotion with smell. So, and that's because of this direct connection through these interneurons of that strong connection between the olfactory receptor cells and the interneurons that are part of the limbic system. Okay, what else to say here? Some fun facts about olfactory reception. Olfactory stimulation is the only sensory stimulation that goes directly to the cerebral cortex without passing through the thalamus. So most other sensory information is routed through the thalamus first and then off to the portion of the cortex that's responsible for interpreting that sensation. It can take as few as 4 molecules of odorant to bring an olfactory receptor cell to threshold and to result in an action potential. So it doesn't take much. We are relatively sensitive, is the bottom line, to odors. But olfactory stimulation adapts very quickly. So after just 1 second, you can see already an increase by 50% of the stimulation that is coming, of the amount of number of the frequency of action potentials drops by 50% after only 1 second of constant stimulation, and it doesn't take long. And so if you've ever been hunting down a gas leak in your house or changed a baby's diaper, then you know that sensation of ooh, that's stinky, or wow, I can't find that gas leak. It disappears very quickly, but it is a central adaptation. And so you can turn it back on again by concentrating and saying, you're telling your brain, I really want to find that gas smell or whatever. We are capable through the olfactory system of distinguishing between 2,000 to 4,000 chemical stimuli. And what's interesting is that multiple receptor types are stimulated by maybe stimulated by a single odorant molecule type. And so it's the pattern of stimulation that arrives at the olfactory cortex that determines how we interpret that smell. So if you turn on, if you turn on a certain group of receptors, then you'll get one smell. If you don't turn on one of those receptors and turn on a different type of receptor, then you're going to get a different smell. If that makes sense. Hopefully that makes sense to you. The book does a pretty good job of explaining that, I think. Okay, so that's all I have to say about olfaction. The next special sense that we're going to talk about is gustation, taste. And taste buds are the sensory organ of gustation. Now, I grew up thinking that a taste bud was one of those bumps there on my tongue. That is not what a taste bud is. So if you'll turn to figure 12.7 in your book, you can see a cartoon there of what a taste bud is. And a taste bud is actually found along the sides of those little bumpy parts of your tongue. So the taste buds are clusters of cells along the margins of those papillae, is what the bumps are called. Okay, so the taste bud is the sensory organ of gustation, and it's built of somewhere between 50 and 100 specialized epithelial cells. And there are two types of cells in a taste bud. There are the taste cells, the receptive cells, the ones that will produce the sensation. And then there are the basal cells, which are supporting surrounding cells. And we find taste buds along, as we said, along the margins of the papillae of the tongue, the bumpy parts of the tongue, as well as up on the roof of the mouth at the soft palate. So if you take your tongue and peel the roof of the mouth, you'll feel the bony parts. I shouldn't try to touch and feel it with my tongue and talk to you at the same time. So you'll feel the bony parts that are anterior. That's the hard palate. And then keep your tongue going back as far as you can along the roof of your mouth. And you'll get to a more muscular or spot where the bone is no longer there. And that bone would, of course, be the maxilla. So that the bone ends and the muscular portion of the palate begins. So that's the soft palate. So we find taste buds scattered across the soft palate. They're also on the inner walls of the cheek and deeper into the pharynx, and also on the epiglottis, the flap of tissue that helps helps direct food toward the posterior side of the pharynx, where the opening into the esophagus is found. So it's like a little ramp that partially covers the opening into the lungs, the trachea. And so it partially covers that and kind of helps the food get to the back side of the pharynx, where it can be swallowed rather than aspirated into the lungs. And so as adults, we have something on the order of 3,000 taste buds scattered throughout the mouth, on the tongue, and then along the walls of the mouth and down into the pharynx. Okay, so let's talk about the taste bud cells now. The first group that we're going to talk about are the gustatory cells, or the taste cells themselves. They have taste hairs on them. So these are slender little microvilli that project from the taste pore that's made by all of the epithelial cells that are around, that surround the taste bud. So they project out into the taste pore and into the fluid medium, which is composed primarily of saliva of the mouth. Now these taste hair membranes, that's where we find the receptor proteins that are embedded in the cell membrane. And sensory neuron dendrites coil up and around these taste cells. And as the taste hairs, the receptor proteins on the taste hairs bind chemicals that are dissolved in saliva. So as they bind them, then they stimulate the sensory neuron dendrites. And that will generate, if there's sufficient stimulation, that will then generate an action potential that will be transmitted into the brain. Taste cells are found in a pretty rough portion of the digestive tract. There's a lot of friction and scraping mechanical forces that damage the cells of the taste bud. And so these taste bud cells are replaced very regularly. Every 7 to 10 days we have a whole new set of taste cells that are there to respond to the foods that we eat. Okay, so the second type of cell that's in the taste bud are the basal cells, and these are stem cells that divide by mitosis and produce new gustatory cells. So they sort of surround the gustatory cells, and as they divide, then they produce new cells that will then differentiate into the gustatory cells. All right, so what are some of the taste sensations and some interesting information about taste sensations? First, sensitivity to taste varies among individuals, but for all individuals it declines as we age. So we have our best taste as young children. And, there's been some suggestion that that's why kids are so sensitive, children are so sensitive to the especially the bitter flavors that we find in vegetables. So Brussels sprouts are off the list for children because they can actually taste a lot of the bitter components of the Brussels sprout flavor. Whereas adults, as we become less sensitive, then the flavor of a Brussels sprout becomes less bitter, less unattractive. And so then it enters the diet. So that change over age is part of what allows adults to eat a lot, stinky cheeses and other materials that children normally, most children, are not attracted to. So taste buds are all physically similar, but despite this superficial similarity, each taste bud is specialized for one of 5 basic taste sensations. There's the sweet. Sweet is stimulated by simple carbohydrates, by alcohols, and sometimes amino acids and lead salts. If hopefully you're not consuming many lead salts, but these amino acids can also provide a sweet taste, and so we use them as low-calorie sweeteners frequently. Lead salts, again, hopefully you're not going to run across those in the environment, but that's part of why paint, flaking paint in old houses with old paint is so dangerous to children, is because if you lick the paint, this lead-based paints, or if you eat the little flakes, they can produce a sweet, that perception of sweet. Sour is the sour taste gustatory cells are stimulated by contact with acids. Remember, acids are chemicals that, when they're dissolved in water, they release a hydrogen ion. So those hydrogen ions help to create that taste of sour. So pickles that have been pickled in vinegar, or fermented foods that have been allowed to ferment with oxygen, they produce lots of acids that give that sour flavor to sauerkraut or to pickles or whatever. Salty flavor taste is stimulated by, now we use the term metal ions, metals in a chemistry sense, so we're not talking about necessarily aluminum, but so sodium, potassium, those are metals that produce this salty not sensation, but perception as we consume them. And sodium by far has the saltiest flavor, and that's why we use sodium chloride to salt our food. That's NaCl. Chloride also provides a little bit of salty stimulation, but not near as much as sodium does. Bitter is stimulated by a class of chemicals that are called alkaloids. So these are alkaline rather than acidic. Nicotine, caffeine, morphine, quinine. So if you've ever had a gin and tonic, part of what gives the tonic water its flavor is quinine. Strychnine. All of these will stimulate a bitter perception. And even some non-alkaloid chemicals like aspirin. Aspirin is actually an acid. It's acetylsalicylic acid, and it has a very bitter flavor. So many of our medications are alkaloids, actually, and so that's why we have the pink stuff. Think about penicillin, or they have a really bitter flavor. And so that's why they are so heavily sweetened, usually with artificial sweeteners. But that's to help overcome that bitter flavor, because sweet kind of helps to negate the taste of bitter, in the same way that bitter negates a little of the taste of sweet. The last of all, back when I was a kid and we were learning about taste, it was only those 4 flavors, sweet, sour, salty, and bitter, that we associated with the tongue and with gustation. But since that time, a fifth flavor sensation has been discovered, or named, maybe is a better way, and that is umami. Umami is stimulated by amino acids and aspartate. That's another chemical that's common in food. And it's what gives that beefy flavor to a steak, or the savory flavor, the yum that you get out of aged cheeses, using MSG, that's monosodium glutamate, that is so prevalent in often in Asian dishes. It gives that the glutamate, which is an amino acid, so it's built off of an amino acid, helps to stimulate those umami receptors. Umami is, I think, it's a Japanese word for tasty. So it's an important flavor. It gives, if you're thinking from a cooking perspective, it gives a lot of depth to the food that you're eating. It helps take a really thin soup, thin tasting soup, to give it a lot more depth and yum to it. Okay, so those are the taste sensations that we can get from the taste buds of the tongue. So why taste? Why is this such an important sense? And it's not just to give pleasure, but it's also a protective sensation. The reason that we notice sour is sour indicates acid. These are acids are generally materials that are going to be damaging to epithelial tissues, the kinds of tissues that line the digestive tract. And so we are way more sensitive to sour than we are to sweet or salty, because it is, from an evolutionary standpoint, a very adaptive thing. We want to notice whether or not there are acids in the food that we're eating, so we can make choices about, do we want to eat this? So, if you are surprised by a real sour flavor, frequently the first response, the reflexive response, is to spit it back out again. So if we are 1,000 times more sensitive to sour than we are to sweet, we're even more sensitive to bitter than we are to sour. 100 times more sensitive to bitter than to sour. So bitter is even more stimulating than sour. And again, it's protective, because so many biological toxins are bitter. That bitter flavor is associated with things that are poisonous. Olfactory receptors, as I was mentioning earlier, also play a very important role in taste perception. So a fully functioning smelling system will really increase taste sensitivity. But with a cold, you lose a lot of that taste ability as well. And you can try out, doing some experiments on this yourself by, eating a piece of onion, or having somebody blindfold you and give you a little piece of onion and a little piece of apple, and see if you can distinguish between the two with your nose held shut, so that you're kind of preventing those odorant molecules from making it up to the smelling parts of the nasal cavities. And it really makes it difficult to distinguish between those two kinds of plant materials. Okay, so how do we get the sensation of taste from the tongue and the cheeks and other places where the taste buds are up to the brain? They're carried on 3 of the cranial nerves. The facial nerves carries input from the anterior 2/3 of the tongue. Glossopharyngeal nerve is what carries the input, the sensations, from the posterior portion of the tongue. And then the vagus nerve carries input from the oral cavity walls and from the pharynx and the epiglottis. So 3 nerves that carry this information. All of them carry the information directly to the medulla oblongata, where the nerve fibers will synapse there with a gustation nucleus. And then the impulses are transmitted up to the thalamus and then on to the primary sensory cortex. So to those regions of the cerebrum there on the posterior, just posterior to that, the lateral sulcus that divides the frontal lobe from the parietal lobes.